Okay, good evening, everybody. I have the honor of welcoming you to the talk and introducing our speaker. So our speaker, who's going to be presenting on neuroplasticity and psychedelics, is Morten Lietz. And he is currently pursuing his PhD, focusing on LSD and neuroplasticity across different ages at the University of Fribourg, Switzerland. Additionally, he serves as a speaker coordinator for ALPS. ALPS is an association that does awareness lectures on psychedelic science and also the yearly ALPS conference. Additionally, he played a key role in initiating research programs at the University of Groningen that combine neuroplasticity-enhancing agents and neuromodulatory treatments for improved outcomes. Another notable achievement, including his co-organization of the first 2 editions of the Summer School on Psychedelic Research with Dr. Brieksum. So without further ado, let's welcome our speaker, Morten Lietz.
Wow. Yeah, thank you so much for showing up. Quite a few people. I was expecting maybe 10, 20. This is amazing. Yeah, first of all, thank you so much to the organizing team for inviting me again, and also for having me to give the same talk again I gave like half a year ago. It's a bit more broader than it used to be half a year ago, so don't worry if you've been here. Yeah, also shout out to the person who did the poster. I think they brought one for me because I liked it so much. Thank you so much. Yeah, about me, so I think the introduction said most of it. So I'm currently doing a PhD in University of Fribourg. I give LSD to old people and measure their neuronal plasticity as a result. It's currently very early, so I'm not going to show you much of the data I have collected there, unfortunately. But I did something in this domain already in the Netherlands, so we will going to talk about that a bit later. But because this is a super complex topic, I would suggest we start at the very basics and yeah, lay out some foundations for also those who are not really into neuroscience to tag along a little bit. All right, so first of all, a bit of definition. What do I mean when I say neuronal plasticity? Well, the definition is the formation of new and alteration of persisting neuronal connections. So basically, neuronal plasticity is this factor that describes how well your brain can adapt to a changing environment. So at a very basic, you've probably heard this term, what fires together, wires together. This is the main concept. There's a lot of different nuance to this, but if we really break it down to the easiest aspect of neuronal plasticity, it means that if 2 neurons fire together, this is a signal to the brain that this is a meaningful connection, and thus it tries to strengthen it. And at the same time, if 2 neurons are not connected in a meaningful way, your brain removes that connection or weakens it. One way to simplify this is by basically thinking about these connections in terms of muscles. So when you train your muscles, that signalizes to your body that you need to grow, that this is important, so you need to grow this tissue, you need to make this connection stronger. Whereas when you have to be at home because you broke your leg or something like that and you can't go out, you're not going to move your leg for a couple of months, you'll see how all these connections, all this tissue basically gets reduced. At the very basics, we can treat neuronal plasticity similar to that. Of course, there's a lot more nuance, it's a lot more complex when we really go into the details, but for the sake of the argument, let's treat it as that, our brain as a muscle. Now, how does it look when we have a look at the synapse itself? So the synapse is the terminal where 2 neurons intersect. So let's see. Oh, can you see that dot? Probably not, it's a bit too weak. Okay, let me do it like this. Yeah. So what happens usually when we look at the synapse in terms of neural plasticity? So there's a signal that's being traveled down through electric signals, which triggers this mechanism where neurotransmitters are being released. They basically work as some sort of key that enters a lock, which is the receptor on the other end of this terminal, and that triggers some sort of reaction. Now, if this happens more often or in a very specific manner that signals the brain this is an important connection, it will make this connection more sensitive. And by sensitive, I mean it increases the amount of neuronal transmitters that is being released, as well as the amount of receptors where these neuronal transmitters attach to, which ultimately leads to a very strong signal in the end. Now, There's also this thing called metaplasticity. Now, this time, because it's a bit more complex than normal plasticity, I'll start with the analogy and then show you how this works in practice. Well, let's go back to this example of training your muscles in the gym. Well, if you would ask Arnold Schwarzenegger, what are the meta factors of your training outcome, he would say healthy food, enough sleep, minimal stress. Now, we can treat neuronal plasticity, metaplasticity of neurons in a similar way. So there are these factors that determine whether your body is currently in the right state to implement these changes. And coincidentally, what's good for the muscles is also good for the brain. So eating enough and eating healthy and sleeping enough and reducing your stress is all a factor that influences your metaplasticity, meaning your potential for change. This is the term that metaplasticity defines. It's the plasticity of plasticity, so your potential to induce neuronal plasticity. And this mostly works via these moderators, or mediators in this case. One, which is quite important, is BDNF and its receptor called TrkB. Now, these two basically, when they are co-activated together with something going on here, it's a signal for the synapse to increase the production of neurotransmitters as well as the production of receptors on the other end. However, if you have more of these receptors in their active state, that means that it's more likely that this change actually will introduce a long-lasting change. Now, let's talk a bit about the role of neuronal plasticity. So, neuronal plasticity plays a role across our whole life. Starting very early on, our brain is very, very plastic because there is just so much unused potential. All the neurons are being formed, all the synapses are already there. However, what's meaningful and what isn't is based on how you live your life. So your brain starts this process of rewiring, of pruning what's unnecessary, of strengthening what is important. Now, this happens in different phases. Sometimes it goes down a bit, then it goes up again when you hit puberty, and then over the course of your life, it will slowly drop down. Because as you can imagine, when we just look at it from a purely evolutionary perspective, at one point your brain is fully matured, and then there's not that much it's supposed to change, right? Because you have adapted to the environment and now go live in it. I don't know if this still makes sense in the environment we currently live in, but back in the Stone Age, this was probably the right play. Right. Oh, nope. Go back. Now, at the same time, for us, this means something quite critical, because when we look at this U shape, this is how normal functioning happens in the adult life. You could put here also neuronal plasticity to some extent, and exchange that for function because it follows a very similar curve. So once you hit a certain point, right around here, which is usually around the age of 55, your brain starts to slowly lose its optimal level of functioning. Now, this is nothing to be alarmed of, because your brain is very well able to compensate for a lot. So you really start to realize, okay, so I start to lose a bit of memory function, I start forgetting things. However, you know how to adapt to that by using techniques, by writing stuff down, because and it's nothing to worry about. However, there's three paths one can take at this point. One, which is the normal path of aging, where you lose some functionality, you retain most of it, and it will stabilize at one point. However, there's also the more cognitive aging part, or the unhealthy part, when you start to develop early symptoms of Alzheimer's. And ultimately, there are very few people who remain very high functioning even throughout age. For a good example, some politicians who still remain in office, I'm not talking about any US politicians here, just to be clear, keep this very high level of cognitive functioning also up until old age. Now, the idea is that whatever determines whether you end up here, or there, might have something to do with how plastic your brain is, so how easy you are able to cope with this change in environment, aka your current brain matter. And one idea is, so why it's so interesting, and why my field also comes into play, the question we had in our lab is, can we get someone who's here at a normal aging process, and can we bring them back up to a higher state of functioning, because we know that they were able to function at that high level at one point in their life. However, more to that at a later stage when I show you a little bit about my study. Right. Of course, not only in your across the lifespan, neuronal plasticity plays a role, but also across a wide variety of disorders. One might say across most disorders. So when we look here, neuronal plasticity has been implemented in degenerative diseases like Alzheimer's, Parkinson's, MS. However, also some very purely neurological disorders, such as hitting your head very hard, so traumatic brain injury, stroke, chronic pain. It has been involved in some neurodevelopmental topics such as OCD and autism, and the one that's most noticeable probably is also in psychiatric disorders such as depression, substance abuse disorders, and schizophrenia. Now, the interesting thing here is when I say has been implicated in, I don't mean it's higher or lower, it's just different. For example, substance abuse disorder usually has been associated with a higher level of neuronal plasticity within certain parts of your brain. Most importantly, those parts that has to do with your reward circuitry. So schizophrenia, very similar, and in autism as well. There's a higher level of neuronal plasticity compared to those who don't fall on the spectrum. So the idea here is that neuronal plasticity is neither good nor bad. Neuronal plasticity is a factor for change, and that can be good or bad. Right, so now the part where you all came for. Let's talk about what does that mean in terms of psychedelics. Well, psychedelics are extremely transdiagnostic, right? So they have been implicated in a bunch of different fields. Let's start at the roots of the current psychedelic research field, which are the clinical trials. The reason why I did this differentiation here is because I think we need to be very careful in what we say psychedelics heal OCD. There's been a study with 9 patients. I think it's a bit too early to tell, but that's what I want to show with this graph. So there are the clinical trials. There have been some success in PTSD using MDMA, but also psilocybin. Psilocybin has also been used to treat addiction. It's also very, very widely used in Geneva, as well as in Klinik Südhang in Bern, to treat addiction. The one that's most popular probably is to use psychedelics to treat depression, but also cluster headaches, end-of-life anxiety. And then, of course, there are the leaves of the psychedelic research field, the more up in the clouds, currently ongoing investigations. So we're looking at eating disorders, chronic pain, dementia, ADHD, and OCD. Now, if there is one substance that does 25,000 different things, one might wonder whether there is some underlying commonality between those different things. And the hypothesis here is that, well, this might be neuronal plasticity. Because neuronal plasticity, if you remember, is this factor for change. And if your brain is in a state in which it is stuck and in which something, this being stuck is what is bothering you, then being able to change that again might bring you back to a more healthy baseline. And that's kind of the hypothesis that has been pushed when it comes to neuronal plasticity and psychedelics. Now, let's look at some mechanisms. So, if you've been at my talk in, I think it was May, you've seen this graph before. So there are many, many different levels at which we can look at more or less the same thing, which is neuronal plasticity. So you can start at the very bottom and you can go up the more you want. Let's start at the very bottom. So of course you can look at neuronal plasticity through just genes that has been associated. For example, the genes that encode certain receptors that we know play a role. You see whether these or not are changing after psychedelics have been implicated. You can also look at the synapse directly through the means of basically just dyeing the receptors or the molecules you're interested in and putting them under a microscope. Cool thing though, you can also use PET. Nowadays there's this one cool tracer where you can mark a protein that is basically ubiquitously layered across all synapses, and you can basically quantify the synaptic density across the tissue, which is pretty cool. You can look at more cells in a culture using like something called voltage clumping. But all of these, I'm not going to go too much into detail because I'm more a person who works with this more higher level ways of looking at neuronal plasticity, for example, using EEG, which is my field mostly. You can also use TMS. You can use network dynamic changes, functional connectivity changes, some other stuff. And of course, you can also use MRI, look at gray and white matter differences before and after certain interventions. So basically, you literally measure how much neurons are there, how much glia is there, do a comparison pre-post. This has all been done. So now, all of these different techniques have been used in psychedelic research so far, some in humans, most in animals, or in cell culture. And then, of course, for the psychologists within you can also look at these behavioral correlates, most importantly, learning and memory, but also cognitive and behavioral flexibility, stress response, all the lot. Yeah. Now, how does it look, again, on a very detailed level? I'm not going to go through that because I barely understand it myself. Luckily, Vollenweider and colleagues have worked out this a bit more simplified graphic. Now, in this case, I'm only going to touch upon the serotonergic effects, the serotonergic psychedelics, because with the others, it gets a lot more complicated. Now, the idea here is that these serotonergic psychedelics, they act as serotonin, and they attach to the serotonin receptors within the brain, right? That means they're agonists, or this is called an agonism. Now, when this happens, it excites the serotonin receptor, which sends out signals. Now, when you are a cortical neuron, you get not only activation through those psychedelics at your own serotonin receptor, but also through these deep cortical layers, which are basically also activated through 5-HT2A. So even though this is not necessarily a serotonin receptor, it still gets triggered through the downstream trigger of those psychedelics. And remember from before, what fires together wires together, and now we have this massive influx of neurotransmitters, this stimulation of this connection, which sends down a signal to, for example, increase BDNF, but also to increase your receptors, increase the neurotransmitter release, and so on and so forth. Now, it also does something in metaplasticity. Now remember, metaplasticity means the modulation of sensitivity. So higher metaplasticity means higher potential for synaptic change. And if you would ask Arnold, or better if you would ask this guy, who knows who that is? I think it's very small, right? Yeah, this is Albert Hoffman. I thought that joke would land better, but now I see it is quite small. He would also say, well, LSD might also have something to do with your metaplasticity. And this is how that works. Now, don't worry, this is a bit complicated, but I will try to make it a bit easier. Now, so we know already that these BDNF receptors, or as I call TrkB receptors, here if we go back one more, these are the receptors that are responsible for facilitating the change, right? So they are not changing themselves, but if there are a lot of BDNF receptors activated through BDNF, this is then the neuron is ready to change, but only if it's now triggered by some kind. So it basically determines whether your brain is currently open for change. Now, the cool thing is that this receptor can have different stages. So it can have the closed state and it can have the open state. Now, if it's closed, it's basically like a clothing pin. And imagine you need to reach the point down here in order to trigger this receptor. But if the clothing pin is closed, it cannot be triggered, it cannot be activated. The BDNF molecule, which is over here, cannot go into this closed receptor. Now, what LSD is doing, it basically, it opens the receptor and then it wedges itself into the mechanism and it keeps it open. And it keeps it open for quite a long time. And now this receptor is primed and ready to go. However, this does not mean that LSD changes your neuronal plasticity, but it changes your metaplasticity, because now these receptors are ready, if activity comes, to also introduce meaningful changes. And this is exactly the point of this paper. So that means if the LSD opens your receptor, but you don't do anything in that state, then there will not be a meaningful change happening within your brain. Now, in other words, if you basically sit around and look for gold in your nose, this will probably not really help you achieve the goals you want to achieve. However, if you use that state, now that your receptors are open and your brain is in a heightened state of metaplasticity, so there is a lot of potential for a positive change, it might be good to start implementing some healthy habits or go to therapy or both. So context is key, that's all I'm saying. Now, when we look at neuronal plasticity over time in terms of psychedelics, there's this one study by Nardu and colleagues. They saw that this is a mice study, which they kind of saw some relations to how long the drug effect is in humans. I don't really know if this is a very valid example, but I think it makes for a nice story, so I keep it in. Take it with a grain of salt, please. So what they saw is they were looking at critical period open states in mice. You don't need to worry too much about what that means. It's a measure of neuronal plasticity in those mice. And they found that there is this really nice correlation between how long these critical periods stay open in comparison to how long the duration of the drug effects are in humans, which I found quite astonishing because that would be quite interesting to know, right? Whether the longer the drug effects, the longer you're in this heightened state of metaplasticity, for example. So just for therapy in particular, this means that if you take, for example, LSD or do LSD therapy, you might have a couple of weeks to actually implement proper change because your brain is now open to implement those changes on a neurological level as well as on a psychological level. Right. Now, let's look at some studies. Let's start at the molecular level. Well, we can look at psychedelics in culture, and even though this is a very cultured psychedelic, this is of course not what I mean. I'm talking about psychedelics in cell culture. Now, very easy thing to do. You get a bunch of cells you want to measure, you put them in a solution. You put this solution, which has all the nutrients those cells need, into an incubator, which is like a reverse fridge, so it keeps your stuff warm. And then these will grow and they will cultivate your plate. And now all you do is you put some LSD or some DMT or whatever the substance of choice is onto those cell cultures and you put them under a microscope and you look what happens. This is exactly what Calvin Lai and his colleagues and their colleagues were doing. This has been quite a benchmark paper because it shows very nicely under the microscope that if you apply drops of these substances into these cell cultures, the tissue was derived from mice and fruit flies, you get these crazy extra branches and there are more terminals and all that stuff. Now, if you have to summarize this means that psychedelics increase your dendritic arbor complexity, so basically if how complex the whole thing looks, but also your spine growth. So a dendritic spine is basically a potential for a synapse to form. It's not a fully formed synapse yet. And lastly, it stimulates the process of turning into a synapse later down the line, if this is a meaningful connection. Again, keep in mind, this has been in cell culture in mice and Drosophila, fruit flies. But cool thing is, very recently, there has been a bit of a breakthrough in terms of using pets and live human beings, not fruit flies, not cell cultures, to actually see whether or not psychedelics do also something to your synapses in the alive human brain. They have been using a PET marker that targets the synaptic vesicle glycoprotein 2A. Just very briefly, a PET marker works you put a label, something that is slightly radioactive onto a molecule that you know is being used in the target area. So in this case, it's like a protein that you need in order to create this SV2A thingy. Now, because you put a radio label on it, which your brain cannot detect, it just simply, you put that into the bloodstream of a person, it reaches their brain, it crosses the blood-brain barrier, and it's being built into those receptors, and after 30 minutes, those start to decay, and you get this radioactive signal, and this is how you make it traceable using PET. And the cool thing is with this SV2A protein, it's extremely ubiquitously and homogeneously located across all synapses of your brain. That means wherever there's a synapse, there's roughly the same amount of this protein. So now if you stick a label to this protein, cool thing is you can basically measure how dense your synapses are across the brain. And this is what they did. Holmes and colleagues, they were using ketamine to see whether this introduces some synaptic density change. And what they found was, well, yes, it does. 24 hours after ketamine, there is an increase in synaptic density across the brain. However, the cool thing or the notable thing of this study was when they further looked at the distribution of the sample, they found that there were half of the people not from the control group, but from the depression sample that had a deficit in these proteins before getting ketamine. Now, those who had a deficit, who then got ketamine, they had this massive increase of synaptic density afterwards, which correlated with a drop in depression symptoms. Those who didn't, so those who started off with a normal amount of synaptic density, they didn't benefit from the ketamine as much, or at all, if you look at this graph. Now, again, very small sample size, so this is like not only a grain of salt, but a good handful of salt. But still, the story it tells here, if being true in a bigger sample, would be that, well, if your depression, which is by itself a very heterogeneous thing to begin with, is not related to your reduced neuronal plasticity, ketamine does not bring the relief you're hoping for. And this points a bit towards the fact that, well, if it's not the neuronal plasticity that the ketamine introduces, then, well, maybe then it's not going to work. Like you need if neuronal plasticity is not the issue, then some drug that introduces neuronal plasticity changes might not be the answer, which I found quite interesting. But again, big grains of salt. Now, something I'm a lot more comfortable with, because this is more my field, let's talk a bit about the network level. Well, so there's this very cool thing there are many different ways of using like different tools on the network level to assess neuronal plasticity. I just handpicked the ones I'm using in my study, which is called sensory-induced LTP. How it works is you put a EEG cap on a person and you give him a stimulus in a not regular frequency, in an irregular frequency. So beep, beep, beep. So it doesn't follow 1 second intervals, but like sometimes it's 1 second, sometimes it's half a second, then it's 0.7 seconds. So your brain cannot predict when the next beep comes. And this gives you those really nice curves in the EEG signal. You can do the same using visual stimulation, by the way. Afterwards, what you do is you now blast that person with the same beep over and over. So beep, 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 beep for a solid 12 minutes. And this signalizes it's kind of crazy. It signalizes the brain, this is important. So let's strengthen the connection that usually is responsible for dealing with that stimulus. And this introduces something called LTP, which stands for long-term potentiation. So basically, your brain becomes more sensitive to that stimulus. And this you do afterwards, you basically do the same as you did before. So again, you do those irregular beeps. And then you end up with these curves. The one in red and green are the baseline, so they did these before. And then the one in blue is the one that you get after you stimulate. And you can see that the brain of these participants actually got more sensitive. So there was a potentiation of roughly 20, well, you can see it over here. There was a potentiation of 0.25. I think these are z-scores. So this is like a normalization. Usually what we see in our study is like 20%. So your brain becomes 20% more sensitive towards this stimulus. Now what you can do is you do the same in a population who does that first, then gets LSD, and then you measure before, after. Or half of the people get LSD, the other half doesn't, or one half gets high-dose LSD, the other one gets low-dose LSD, and that way you can compare these very tiny potentiations. And what we see, I can give you a bit of a preview in the study in young people, which is almost finished. LSD adds roughly 20% to this whole potentiation compared to... So what we want to see is that everyone potentiates, even those who have... nice. Even those who who didn't get any LSD or who were just about to get LSD, but the ones who did get LSD, they potentiate even more. That is the whole idea. There was this group who tried to do exactly this method in a depression sample using psilocybin. They had 19 patients, so also was quite small. They had to be off medication because especially SSRIs also introduce neuronal plasticity changes, and they gave a medium dose of psilocybin. And they used the same paradigm I just described. So this is what they found and what they proudly showed the world, and it's a bit weird because for those of you who I didn't lose in the last couple of slides, you will already see that, pre-tetanus, tetanus basically means the stimulation part. You can see here that when they did it like at placebo, so there was nothing given, before the stimulation is roughly the same as after the stimulation, so no potentiation happened. Now, this could mean that you cannot potentiate in a depressed brain, or it means that your method doesn't work. Because if there's no potentiation happening, then you can't measure this neuronal plasticity, because the potentiation is what you want to measure. And so they, after 24 hours after psilocybin, they try to do the same, but they have the same result. Then 2 weeks after, it looked a bit better, but also not significant. And then 2 weeks after psilocybin, you see this massive increase suddenly. However, again, pre-stimulation and post-stimulation is still the same. Now, this is a bit weird because, first of all, they use theta power, which is already a further advanced technique. When you look at these EEG signals, talking from experience, when you work long enough with EEG, you will find something. And what you can do is, if the ERP signal doesn't show what you want to show, what you want to see, then you basically just keep on digging and you look at the different frequencies, and maybe one of them does something that follows your hypothesis more or less. And they did not justify that. And when I contacted them, they didn't reply. So because I was a bit, I thought first of all, hey, that's super cool, but ultimately they want to measure metaplasticity, and this is not metaplasticity. This is something else. So I don't know. Let's take that with a grain of salt again. And however, what they did see was that those who got psilocybin got reduction in depression scores, but this might be unrelated to the neuronal plasticity they thought they were measuring, because this is what we see in other studies as well. Yeah, and I already skipped ahead, so this is what I meant with being careful looking at these results. Right, for those psychologists among you, let's look at the behavioral level. So there's no very clear-cut correlation where we can say, yeah, it's neuronal plasticity and this function is definitely the same thing measured from a different way, so it's still a bit vague, but most people would argue that psychological flexibility, so basically how your behavior is flexible, is a measure, or is at least a correlate of neuronal plasticity. This was the same group, by the way, and they found that after psilocybin, your inflexibility score, so in other words, your flexibility went up, inflexibility went down, and this persisted for 3 months. However, they brought this out in a new paper, and they didn't correlate that with the EEG paper, and they didn't mention that these two were from the same study, so that's another reason why I was a bit like, I don't know. Luckily, there was another study by Davis and al. Who found that increase in psychological flexibility is basically the mediator of decrease in depression and anxiety, which mediates between insights and mystical experiences. So I thought that was actually better evidence for me to say, yeah, probably psychological flexibility plays a major role in this demand, in this model. Of course, there's also cognitive flexibility. This you can measure using, for example, the Penn conditional exclusion task, which looks like this. The task here is very simple. Depending on what rule is currently active, you need to decide which one is the odd one out. When we look at the one on the top, if the rule is look at the color, then this one would be the odd one out. If the rule is look at the orientation, then this one would be the odd one out. And that changes rapidly throughout the trial, so you always have to flexibly implement another rule set in order to still give the right response. And so what they found is in those 24 patients with MDD, after 2 sessions of psilocybin, that their cognitive flexibility was improved because the errors they made were less. Now, This, interestingly enough, did not correlate with their reduction in depression score, because as you can see here in the very top, they did have less severe depression symptoms, which I thought was actually a good sign, because if this is independent from their decrease in depression, it means that the cognitive flexibility was not impaired to begin with because of their depression. But now this it tells a bit, it shows a bit more that this might be due to this neuroplasticity effect from the psychedelic itself. But again, very, very early. It's way too early to tell for certain. Right. So how can we use this in practice? Well, the idea here is that let's imagine that your brain is a pot where you cook rice in, and you are a very forgetful person, and you left it on for too long, and now you have this big mess where there's a lot of sticky rice at the bottom of your pot, and you really have to scrape really, really hard to get it off. Now, in this scenario, let's imagine your like the rice that is very sticky is your mental illness, and the sponge you use is going to therapy. You can go scrub away, no problem, you will get there. However, your brain or the pot is not very receptive. It's not in the most optimal state of change. Now, the idea is if we find a way to introduce dish soap and warm water and let it sit for a while, maybe this will improve our likelihood to actually introduce meaningful change using the intervention at hand. And this was roughly the idea I had very, very early on. Way too early if you ask me, and we just kind of went with it. And this is how this project came to be, which I'm now going to show you some data. It was recently published. So this is the project I was talking about earlier, which I did in the University of Groningen, where we try to use microdosing psilocybin in order to improve neurofeedback, which is a technique that is also based on neuronal plasticity. The idea here was to use the psilocybin to put the brain in a more receptive state, and then the neurofeedback to guide the brain towards meaningful positive change. Many thanks to the people who work there, most importantly Stephanie Enrique Skeppert, who was my mentor all the way throughout, and she now just got a kid, and we're all really happy. But that means that I'm on my own now in this domain. So there's a link to the paper down here. I can put it up later, sorry. So let's start. What is neurofeedback? So neurofeedback is a technique where you can use fMRI, but you can also use EEG. We used EEG because it's easier. What you tell the people. Let's start again. So there are certain underlying networks that are coupled to certain functions. So, for example, executive functions like planning, decision-making, all that stuff is all based on network activity of your executive function network. And the cool thing is that this network you can measure very well right here at the very top of your head using EEG. And what we know is if there is a lot of activity within that network, that means that you perform really well at these tasks. And if there is not enough, then you perform poorly. And well, if I tell you now try to activate that network, you will probably not be able to do that, because that's very hard, because you don't know what you need to do in order to activate certain parts of your brain, right? Well, this is where neurofeedback comes into place, because what it does, it reads out the network activity within that specific network. It feeds them through the computer, and the computer gives you some sort of feedback that tells you now you're using your network, now you're actually not using your network, or now you're upregulating, now you're downregulating, and it gives you feedback in order to help you keep using that network. And if we go back to one of the earlier slides, remember the muscle and the brain, right? If you use a network, it means you stimulate growth within that network. And the idea here was to use psilocybin in order to boost that. But let's first look how that looks in practice. Nope. And play. Does that play? Yeah. Well, I mean, it's not that interesting anyway. So as you can see, it's kind of boring. It's a square. There's a lot of technological advancement to make a square go from gray to red, which is increased activity, to blue, which is decreased activity. Doesn't really matter. But this is how it looks like. There's a lot more cool stuff you can do with it. This is the more simpler ways which we used. And the idea here was to have one group who gets to microdose for a week and then another week in combination with neurofeedback, 3 sessions, and compare that to a passive control group who had to wait for 2 weeks and then they started microdose. We measured different things. For example, we had some tasks that looked at executive functions, but also some functionality scores, mostly via questionnaires. And one thing which I find very important here to mention is that this was a proof of principle study because we were completely unfunded. So the study design is crap. This does not get rid of any expectancy effects. This does not get rid of any placebo effects. And most importantly, this does not show that psilocybin with neurofeedback is better than neurofeedback on its own. But this was the only thing we could do in order to set out on this journey. And then later on, we would use this data in order to write grants and see if we can get someone to fund this. So far, without success. But let's see. Let's look at some data. So first things first, it has potential. So we managed to increase the theta amplitude, which is this activity within that executive function network. And in the first session, they started off here and they ended off here. You see they were struggling a bit. In the second session, they managed and then went back down again. And then in the third session, there was some sustained growth happening. And this is how it looks like on average. So yeah, kind of okay. So we did manage to upregulate activity within that network. Big question remains, is this better than without psilocybin? Did the psilocybin add anything? With this data, we cannot tell. But what we can tell is that people managed to pull it off and nobody died and everyone was happy. And this is a lot more important than you would think, but a lot of people were asking us exactly those questions when we were applying for grant money. So yeah, we could tell them, no, everyone was fine, which was nice, and people liked it. So, but yeah, then when we look at the behavioral data, you can see that there is a bit of a slight improvement in those who did get the treatment, but nothing very significant. So that was a bit of a sobering experience. The question is, maybe this is not sensitive enough. Maybe we didn't have a high enough dose of neurofeedback sessions, because usually what's common is that you give like 5 to 8 sessions of neurofeedback. We only did 3 because you can fit 3 in a week. That was a bit more manageable. But yeah. And then, of course, when you ask the people, so do you think you got better? Everyone said, fuck yeah. Because psychedelics tend to do that, and the people who get neurofeedback tend to do that as well. So these two populations together, the ones who like neurofeedback and psychedelics, they're quite suggestible, one might be able to say. Or this is the breakthrough we were hoping for, but I'm a bit more skeptical. Yeah, and then when we look down here, when we ask them what their goals were and whether they have achieved them, they also they said that, yeah, they were expecting a lot of achievement, and they actually got a lot better in their cognition as well as presence and mood. But then again, these are very subjective ways of asking, and ultimately all we wanted to show is, does this work at all? Is this worth pursuing further? I would say yes, but probably not the way we did it. There need to be a lot of adjustment in the protocol later on. However, this project was great because this was my very first research project. I was completely in over my head. And I learned a lot from these. Most importantly, there are a lot of open questions that remain, mostly the translatability, because a lot of the very cool results I've shown you so far were all from animal studies, and we have no idea whether this are actually true in humans. Then the whole question whether upregulation versus normalization actually works. So can we actually increase neuronal plasticity in healthy young patients where neuronal plasticity is roughly at an optimum level? The reason why we are wondering this is because there's a lot of reasons for your brain to downregulate if you get too high, because this is also a sign of, not optimal level of functioning. As you remember from before, certain disorders are based on too high neuronal plasticity, so your brain has protective mechanisms. So importantly, this becomes quite interesting if we look at elderly studies, what I'm doing currently, because there we know neuronal plasticity goes down in old age. Maybe we can bring it to a prior level. Maybe we can't infinitely boost that, but maybe we can, go like a healthy aging approach. That would be cool. Then, of course, there's the question of chicken or the egg. So does psychedelics induce neuronal plasticity, which then lead to positive change? Or is it the other way around? Because the psychological experience leads to the person behaving in a more positive way, in a more different way than before, and that is your brain answers to this change in behavior with increase in neuroplasticity. We don't know. And last but not least, how long, if this all works out, if we see that there is a neuronal plasticity change, how long can we actually make use of this? Is this long enough for us to actively introduce some positive change into our lives, or is this just over within basically the psychedelic experience itself, and then the days after you're not even that increase in neuronal plasticity? Well, I try to answer a few questions with the LINES project, which does not stand for what you probably think it does. It's for LSD-induced neuronal plasticity in elderly subjects, and the elderly only to fit the E. 55 and older are not elderly, but just it's an older adult population. And these are our three main goals: neuronal plasticity, cognitive performance, and safety, because so far there's been only, I think it was there's a study that showed that 1.2% of all study participants were 65 years and older. So we have very, very few data on this population. And besides doing all these neuronal plasticity assessment and cognitive assessment, I also take a load of safety data while doing so. So this is all quite nice, because then we can also give LSD to old people and people above 55 and not worry too much. Right. I was already worried that I'm going a lot over time, but this is fine. Bit of advertisement. So if you want to participate in my study, we're definitely still recruiting throughout the whole of 2025. So if you're not 55 or older, that's completely fine. You can, of course, show your parents or grandparents or whoever else is 55 in your age. Yeah, so there's a link to our website where you can get more information. The QR code will get you there as well. And because academia is difficult on your own, we also need interns every now and then. And I still have some slots available starting 2025 or 2026. So if you know how to work with EEG or did some work with participants already, yeah, hit me up. Cool. And this is how I feel every day in this job. And thank you so much.
Okay, thank you so much, Morten. We'll now be taking your questions, so just raise your hand and we'll come to you with a microphone.
Thank you. At the beginning, you spoke about the correlation between neuroplasticity and some conditions like OCD and others. Do you know the correlation between neuroplasticity and color blindness?
Color blindness?
Yeah.
Color blindness is can have many different reasons, right? So there's like an issue within the eye. There might be an issue within the brain. So I am not aware that it has to do with neuronal plasticity in a way, unfortunately. But I'm also I know too little to give you a solid answer.
Okay, it's fine. Thank you.
Thanks for the talk. In the beginning, you mentioned at this lower level, right, that the density of synapses has changed. So I just wanted to get a better understanding how expensive it is for the brain to increase or decrease the synapse density. What's the lifetime of that change, and what's the main catalyst for that?
Oh, those are very good questions. Let's see. So this study has only looked at 24 hours post-change, so we don't know for certain. However, there were some studies that looked at functional connectivity change, so basically how your brain communicates over time. And they found that after psilocybin administration, 3 months later, these functional connectivity changes were still increased in a depression sample. So it seems to be once that change, this upregulation of synapses occurs, your brain starts to decrease it in those areas where it's not meaningful, but it keeps it there where it is meaningful. And then these changes you can see quite a long time later. Now, this is not directly something that is neuronal plasticity per se, but this is just a result of neuronal plasticity, right? Because what we would expect, also based on the animal studies, is that this window closes after a week, maybe 2. Yeah, and what were the other questions?
What's the main catalyst? But is that what you mentioned, this opened receptor that increases the plasticity, right?
We don't fully understand. What means we not fully? We barely understand this mechanism. Let's start off with that. So we have a good idea that BDNF plays a role. So BDNF is one of these metaplastic hormones in your brain that, if it is there in abundance and if the receptor is in a state in which it is open, it means your brain is more easily malleable, so it's changing more likely. But of course, there's also something has something to do with your NMDA receptors. It has some stuff has to do with your genes. So how there are different subtypes of genes that determine how much the abundance of this receptor is. So there's a lot, and we're only at the very early stage at understanding this super complex topic.
Okay, thank you.
You mentioned, or you said, neuroplasticity is a factor of change. In general, in my studies, and when I hear about neuroplasticity, it's a word I have a great difficulty defining concretely. Or
Me too.
It's because in the way that I think about synapses, there's always activity, there's always change. So how do you do it? When does changes in LTP or changes in synaptic connection reach the point where it can be called neuroplasticity? Or is it just a broad term that is used generally?
So let's see. Because basically the question almost boils down to what's the difference between plasticity and metaplasticity, right? Because I always have the difficulty of keeping those two apart because they're so closely related that I also think they're both necessary for neuronal plasticity to occur. But let me try to phrase it this way. If you use TMS, like a transcranial magnetic brain stimulator, and you stimulate the motor cortex and you measure the outcome on the muscle, if you stimulate that over time and quite frequently, there will be a change in excitability happening within that connection. So it will either go up or it will go down, and that you can measure. That would be something called hyperplasticity. So basically excitability, that is not directly the plasticity that we try to measure, because what we try to measure is the potential for change and not the change itself, right? Now, if you do this in the morning, and then you do that again in the afternoon, or before and after LSD, you see how that change happens over time, and that's more like the metaplasticity. No, it's also not true. So I'm having a lot of difficulty answering that question because without BDNF action, there's also probably no LTP change. But so it is very, very difficult. Yeah, and I do agree, and I do have difficulty keeping those two apart as well. Yeah, but let's try together later, because now I already started down a path and I'm a bit nervous. And yeah, but very good question. It's hard. It's a really hard topic.
So you mentioned SSRIs, and that they also have a mechanism of action that may be related to neuroplasticity. Do you happen to know if the mechanism of SSRIs and psychedelics on neuroplasticity are anyhow similar?
So the idea is that they probably are to some extent. Everyone thinks it most likely has to do with their serotonergic action. I'm talking SSRIs here in particular, which is this drug where it basically keeps your serotonin within the active state so that it doesn't get put back in storage, but active over time. However, we don't even know whether this is the main action of SSRIs to begin with, because SSRIs are also not fully understood yet. One interesting thing, though, is that the neuronal plasticity seems to be upregulated in SSRIs roughly around the same time where you have the first positive effects from the treatment, which is after 6 weeks of taking them continuously. However, you do see serotonergic action basically immediately after intake, so 30 minutes after you have all the serotonergic stuff, but the neuronal plasticity change happens after a prolonged period of time. So here it seems a lot more the case that because you get better, because there's this therapeutic outcome, your brain reacts to that by normalizing your neuronal plasticity in a more endogenous way, whereas with the psychedelics, this happens within 30 minutes, and it happens a bit more violently. So here, it looks a bit more that this might be because they do exogenously, so by themselves bring these neuroplastic changes.
Okay, thank you.
So you mentioned microdosing, and I was wondering regarding healthy people for like improving habits, for example, if there is any evidence that if you, for example, microdose with psilocybin, if there's any evidence that this actually helps to improve your habits, or if it's like the placebo effect, like you said, like the metaplasticity, that you just choose to engage in these habits more because you like chose to do it because of you took the psychedelic.
Yeah, that's a great question. I mean, coming from a microdosing focused angle myself, that was exactly what we were wondering, even though we weren't directly measuring it, right? Because we didn't measure neuronal plasticity, we measured the effects of neurofeedback combined with blah. So, so far there were a couple of studies that looked whether microdosing on their own was actually changing something. Let me see if I... Oh, Jesus, no, we're not doing that. Because I had a slide on this. There was one microdosing study. I think I took it out, so I'm not sure. Yeah, nope, I took it out, sorry. There was a group that looked at... Oh, I fucked it up. Ah, doesn't matter. There was a group that looked at the effects of microdosing, and when I say microdosing here, I mean the Maastricht definition of microdosing, which is 24 micrograms, which I don't know if those of you who ever microdosed, I think 24, that's a bit high. I wouldn't go to work with that. I don't go to work with microdosing anyways, but generally I would not recommend. That's a museum dose in my opinion. But okay, opinion over. They know what they're doing. They're great guys. So they used quite a high dose. We expect that the neuroplastic effects are dose dependent. So the higher the dose, the more strong the effects. They used 70 people. And they used only male participants because they were afraid of hormonal changes. So the signal-to-noise ratio was basically close to optimal in that regard. And they didn't find search. So it didn't work. The whole paradigm didn't work. They couldn't induce. They used a similar paradigm that I was describing with the EEG setup and the stimulation. They had 70 people, and it was inconclusive in the end. There was another study that looked at BDNF biomarkers in the blood. However, it turns out BDNF is probably bullshit when you measure in the periphery because it does not reflect what's going on in the brain. So as of now, the state of knowledge is that we don't think that microdosing meaningfully, or at least with the tools that we have in order to measure increases in neuronal plasticity. Whether or not this is also true for cognitive flexibility or behavioral plasticity is another completely other question, which is still up for debate. Ultimately, if it helps you, whether or not it's placebo, do with that what you may.
Thank you.
Okay, with the sound of the bell, I think that concludes tonight. A closing remark. For some people might have expected a talk on creativity tonight. For those of you that didn't hear, our speaker had to cancel, or rather postpone for next semester. So for those of you interested in learning more about the effects of psychedelics on creativity, we have a fantastic speaker who did research at the Psychiatric University Hospital in Zurich, presenting that talk next semester, so that's already an outlook. And thanks to Morten for being so flexible to jump in and present on the topic of neuroplasticity. Thank you all for coming all the way from Fribourg. So give up a last round of applause for Morten.
Thank you guys. Thanks for having me.
Before we release you into the uproar, which is right next door, an outlook for soon. Well, I don't have my laptop. So next week is the last talk of this semester, which is going to conclude the talk series for this semester. And in 2 weeks. It's going to happen at the University of Zurich in the main building. It's going to be by a psychotherapist, so he's a licensed psychotherapist in Switzerland, and he is working with patients currently with various diagnoses using psychedelics to treat these symptoms. So he's currently administrating psychedelics to people legally and using them to treat symptoms in the context of psychotherapy and using them as a tool in psychotherapy. So if you're interested in learning more about work with patients and how psychedelics can be used to treat various disorders, you should come by. He is a fantastic speaker, very passionate in this field, and you'll also have the opportunity to ask your questions and get to know him. So we look forward to welcome you all, and that will conclude this semester, and then we're ready in the planning of next semester, so this is going to keep going. And yeah, thank you all for coming, and have a lovely evening.
So in case you know someone who wants to participate in my study, I have flyers. I put them out here. Otherwise, I would regret not using this opportunity.
That's it. Thank you. Thanks.